In this video, we will talk about the integrated functions of the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase of digestion is the gastric secretion that occurs even before food enters the oral cavity. Smelling, seeing, or even thinking about food can kickstart the cephalic phase of digestion. Anticipatory stimuli and the stimulus of food in the mouth activates neurons in the medulla oblongata. The medulla then sends an efferent signal through autonomic nervous neurons to the salivary glands and through the vagus nerve to the enteric nervous system. In response to these signals, the stomach, intestine, and accessory glandular organs begin secretion and increase motility in anticipation of the food. The first step is the chemical and mechanical digestion that occurs in the mouth. There are three kinds of salivary glands: parotid salivary, sublingual salivary gland, and submandibular salivary gland. The main secretion of the oral cavity is saliva, which has four important functions. Saliva softens and moistens food, lubricating it and making it easier to swallow. Saliva digests starch using salivary amylase. Amylase breaks starch into maltose after the enzyme is activated by saliva. Saliva dissolves food so we can taste it. Saliva also acts as defense, disabling bacteria and viruses using lysosomes and immunoglobulins. Saliva also helps to wash the teeth and keep the tongue free of food. The mechanical digestion of food is called mastication, resulting in a softened, moistened mass called a bolus that can be easily swallowed. Saliva is a complex hypoosmotic fluid that contains water, ions, mucus, and proteins such as enzymes and immunoglobulins. Salivary glands are exocrine glands with secretory epithelium arranged in clusters of cells called acini. Each acinus surrounds a duct, and the individual ducts join to form larger and larger ducts. The main excretory duct of each gland empties into the mouth. Salivation is under autonomic control and can be triggered by sight, smell, touch, and even thought of food. Parasympathetic innervation is the primary stimulus for secretion of saliva, but there is also some sympathetic innervation to the glands. The food is then swallowed using a reflex action that pushes the bolus into the esophagus. The esophagus leads into the stomach, which brings us to the integrated function of the gastric phase. The stomach has three general functions, the first being storage. The stomach stores food and regulates the food to enter the small intestine. The second function is digestion, as the stomach is where chemical and mechanical digestion occurs, turning the food into a soupy mixture called chyme. The third function is defense. The stomach offers the body protection by destroying many of the bacteria and other pathogens that are swallowed with food or trapped in airway mucus. There are a number of reflexes that aid the gastric phase. Distension of the stomach and presence of peptides or amino acids in the lumen activate endocrine cells and enteric neurons. Hormones, neurotransmitters, and paracrine molecules then influence motility and secretion. The stomach has deep gastric pits that lead to gastric glands deep in the mucosal layer. G cells are found deep in the gastric glands. The G cells secrete the hormone gastrin into the blood. Gastrin release is stimulated by the presence of amino acids and peptides in the stomach and by distension of the stomach. Neural reflexes also trigger gastrin release. Short reflexes are mediated by an ENS neurotransmitter called gastrin-releasing peptide, or GRP. Gastrin promotes acid release by acting on parietal cells and indirectly stimulating histamine release. The parietal cells secrete gastric acid, also known as HCl, into the lumen of the stomach. The stomach produces two enzymes, pepsin and gastric lipase. Pepsin carries out the initial digestion of proteins. It is very effective on collagen and thus is helpful for digesting meat. Pepsin is secreted by chief cells in its inactive form, pepsinogen. Once in the stomach lumen, pepsinogen is cleaved to active pepsin by hydrogen protons, and protein digestion begins. Gastric lipase is co-secreted with pepsin. Lipases are enzymes that break down into triglycerides. There are also many paracrine secretions at work. Paracrine secretions from the gastric mucosa include histamine, somatostatin, and intrinsic factor. Histamine is a paracrine signal secreted by enterochromaffin-like cells, ECL cells, in response to gastrin or acetylcholine stimulation. Histamine diffuses to its target, the parietal cells, and stimulates acid secretion by combining with H2O on parietal cells. Intrinsic factor is a protein secreted by the same gastric parietal cells that secrete acid. In the lumen of the stomach, intrinsic factor complexes with vitamin B12, a step that is needed for the vitamin's absorption in the intestine. Somatostatin, also known as hypothalamic growth hormone inhibiting hormone, is secreted by D cells in the stomach. Somatostatin is the primary negative feedback signal for gastric phase secretion. It shuts down acid secretion directly and indirectly by decreasing gastrin and histamine secretion. Somatostatin also inhibits pepsinogen secretion. The stomach balances digestion and defense. The gastric mucosa of the stomach protects itself from autodigestion by acid and enzymes with a mucus bicarbonate barrier. Mucus cells on the luminal surface and in the neck of gastric glands secrete both substances. The surface of the intestinal epithelium is called the brush border, from the bristle-like appearance of the microvilli. There are many intestinal secretions that promote digestion. Secretions include digestive enzymes, bile, bicarbonate, mucus, and an isotonic sodium chloride solution. Digestive enzymes are produced by the intestinal epithelium and the exocrine pancreas. Intestinal brush border enzymes are anchored to the luminal cell membranes and are not swept out of the small intestine as chyme is propelled forward. The control pathways for enzyme release vary, but include a variety of neural, hormonal, and paracrine signals. Usually, stimulation of parasympathetic neurons in the vagus nerve enhances enzyme secretion. The liver produces bile, which is then released into the gallbladder. Bile is a non-enzymatic solution that allows for the digestion of fats. The small intestine secretes bicarbonate secretion into the small intestine, neutralizing the highly acidic chyme that enters from the stomach. Bicarbonate is released in response to neurostimuli and comes from the pancreas. The intestinal goblet cells produces mucus, which protects the epithelium and lubricates the gut's contents. An isotonic sodium chloride solution secreted by the crypt cells in the small intestine and colon mixed with mucus to help lubricate the contents of the gut. The pancreas secretes enzymes and bicarbonate. The pancreas is both endocrine and exocrine. Endocrine secretions come from clusters of cells called islets and include the hormones insulin and glucagon. Exocrine secretions include digestive enzymes and a watery solution of sodium bicarbonate. Ducts of the acini empty into the duodenum. The acinar cells secrete digestive enzymes and the duct cells secrete the sodium bicarbonate solution. Pancreatic enzymes must be activated when entering the intestine. Enteropeptidase converts inactive trypsinogen to active trypsin. Trypsin then converts the other pancreatic cyanogens to their active forms. The signals for pancreatic enzyme release include distension of the small intestine, the presence of food in the intestine, neural signals, and the GI hormone CCK. Pancreatic enzymes enter the intestine in a watery fluid that also contains bicarbonate. Bicarbonate secretion into the duodenum neutralizes acid entering from the stomach. A small amount of bicarbonate is secreted by duodenal cells, but most comes from the pancreas. The liver secretes bile, which is a non-enzymatic solution secreted from hepatocytes, which are liver cells. Bile consists of bile salts, which facilitate enzymatic fat digestion, bile pigments such as bilirubin, which are the waste products of hemoglobin degradation, and lastly cholesterol, which is excreted in the feces. Bile is secreted to the gallbladder, which stores and concentrates the bile solution. Most of the digestion occurs in the small intestine. Enzymatic fat digestion is carried out by lipases, enzymes that remove 2 fatty acids from each triglyceride molecule. The result is 1 monoglyceride and 2 free fatty acids. Fat digestion also requires colipase, which is a protein cofactor secreted by the pancreas. Colipase displaces some bile salts, allowing lipase access to fats inside the bile salt coating. Phospholipids are digested by pancreatic phospholipase. Free cholesterol is not digested and is absorbed intact. Carbohydrates are absorbed as monosaccharides, broken down by amylase in the mouth and pancreatic amylase. making starch into maltose. Maltose and other disaccharides are broken down by brush border enzymes known as disaccharides.